Carbohydrates. These are a major source of energy that we know of in our diet. Just like the name tells you, they are carbo, means they consist of carbon. Hydrate essentially means they consist of water. So a general formula of a carbohydrate is essentially CnH2ON, which means if you have n number of carbon atoms, you have 2n number of hydrogens and n number of oxygen. So that is why they are called as carbohydrate. They are made up of carbon, hydrogen, and oxygen majorly. There are some other names that are used for carbohydrates. For example, sugars is a commonly used names. All the kind of sugars that we consume, they're usually carbohydrates. Another commonly used name is called saccharide. So if you ever hear saccharides, monosaccharides, disaccharides, we are majorly talking about carbohydrates here. One example of carbohydrate, common carbohydrate is glucose. How are these carbohydrates usually produced? So we all know carbohydrates basically are the sugars that are coming from plants. So they are usually synthesized by plants through the process of photosynthesis, in which CO2 and water is used and energy from the sun is used to prepare sugars that are called as carbohydrates, which are then consumed by other organisms, and they are used as a source of energy. The process by which these carbohydrates are broken down back into CO2 and energy is called respiration. So the two processes are basically either a synthesis of carbs or use of carbs, or photosynthesis and respiration. One is CO2 and water gives you carbohydrate, which is glucose. Another one is glucose is broken down in presence of oxygen to give you CO2 and water, that is respiration. Okay, so let's talk about what are different types of carbohydrate. Carbohydrates, you could classify them into three major classes. Like I said, another term used for carbs is saccharides. So you have something that is called as monosaccharides, which is the simplest type of carbohydrate. So these are the simplest sugars, monosaccharides. When two monosaccharides combine with each other, they give you something that is called as a disaccharide. For example, this is one simple sugar. When two of these sugars combine, they give you what is called as disaccharide. So when two monosaccharides join, when multiple of these monosaccharides join with each other, they give you something that is called as polysaccharides. So essentially, when a monosaccharide is digested, you cannot further go any down than that. It will be broken down essentially into CO2. Okay, if you break this further down. When you break the disaccharides down, you could break this disaccharide in the middle, and that would give you two monosaccharides. When you break these polysaccharides, then that would give you multiple monosaccharides. And this process of breakdown of a sugar is essentially usually called as hydrolysis. Breaking, lysis again means break, hydro means in presence of water. So hydrolysis is breakdown of something in presence of water. Talk about what are these carbohydrates structurally. So we'll start with monosaccharides, which is the simplest unit of a sugar. So monosaccharides would consist of a chain of carbons. Like we know, it's an organic compound, so it's basically a chain of carbons. Monosaccharides are made up of anything between 3 to 7 carbon atoms. What are these? These 3 to 7 carbon atoms. For example, if I draw a 4 carbon compound like this, these are my 4 carbon atoms. They will be connected to each other. They will always consist of one carbonyl group, and they will always consist of hydroxyl groups on all the carbons other than the carbonyl group. So if you remember, carbonyl is C double bond O group. And there are two types of carbonyl. One is called an aldehyde group, which is if you have a C double bond O, one is connected to a carbon, the other one is hydrogen. This is called an aldehyde group. And the second one, if you remember, is a ketone group, where you have a carbonyl group connected to carbon on both the sides. So this is a ketone group, and this is aldehyde. CHO or C double bond O. That is what makes two different types of monosaccharides. If a monosaccharide or a sugar consists of an aldehyde group, it is called as an aldoses sugar. If it contains a ketone group, it is called as ketosis. And apart from this carbonyl carbon, every other carbon has a hydroxyl group attached to it. They have hydroxyl groups on all carbons except carbonyl. So for example, if I want to draw 4 carbon carbohydrate, how would you draw it? Very simple. You know it has to be a ketone or an aldehyde. So if it's a ketone, it will be here, CHO, CH, I'll put a double bond O right here. Okay, so this is aldehyde group, which means the other carbons have to have at least one OH. So I'll put an OH on all. Then you satisfy the rest. So this carbon already has 2, 3, and 4 bonds, so it does not need any more. This has 1, 2, 3 bonds, so it needs one more, so I put a hydrogen. This carbon has 1, 2, 3, so it needs one more hydrogen. 1, 2, which means it needs 2 more hydrogen. So that's how you usually draw structure of a monosaccharide unit. This, for example, is showing you this is a 4 carbon, 1, 2, 3, 4, 4 carbon aldose sugar. Why? Because it has an aldehyde group. So this is called as erythrose. 1 carbon, 2 carbon, 3 and 4 all have OH, except there's one carbon which has a carbonyl group, C double bond. Similarly, you could have a 4 carbon ketose, which is called as erythrulose, which is 1, 2, 3, 4 carbons, and it has a ketone group. Remember, ketone has to have 2 carbons attached to it. That is why the second carbon is always going to be the ketone carbon. The first carbon, aldehyde, will always be the first carbon, ketone will always be the second carbon. And the rest, every single carbon has OH. So it's very simple to draw structure of a carbohydrate. Every single carbon should have an OH except the carbonyl. Based on how many carbon atoms are present in a carbohydrate or in a monosaccharide, you could classify it. For example, if it has 3 carbons, it's called a triose. 4 carbon is called as a tetrose, 5 carbon is called pentose, and 6 carbon is called hexose. So based on the number of carbon atoms, you can classify the monosaccharides as well. So one is classification is if it has aldehyde group, it is called aldose. And if it has a ketone group, it is called a ketose. And second classification is based on the number of carbon atoms. So for example, if we say you have an aldopentose sugar, that means you have a pentose, means you have a 5 carbon sugar, which aldo means it has an aldehyde. So aldopentose means it's a 5 carbon with aldehyde group. Ketohexose would be a carbohydrate which has 6 carbons. Hexose means 6 carbon. Keto means it has a ketone group, or you can say it's a ketosis sugar. So a ketohexose is a 6 carbon monosaccharide with a ketone group. So you might see these names: aldocrete, ketose, aldopentose, aldotetrose, aldohexose. Those just tell you what number of carbon atoms and what type of carbonyl group is present in that monosaccharide. Here you see some examples of monosaccharides. For example, this is a 3 carbon monosaccharide with an aldehyde. So this is called aldotriose. Example is the actual name is glyceraldehyde. This is 1, 2, 3, 4 carbon, and this is called as aldotetrose with an aldehyde, triose. Aldopentose, so 1, 2, 3, 4, 5 carbons and aldehyde, so that is why it is called aldopentose. Name is ribose. Fructose, which you might have heard of a lot, fructose sugar. This is a ketohexose sugar, which means it is 6 carbon, 1, 2, 3, 4, 5, 6 hexose, and it has a ketone group. That is why this is ketohexose. So this is fructose. Now, somebody might say that, okay, we just saw a 4 carbon sugar with an aldehyde in the previous slide, and we said that that was erythrulose. Whereas now we are saying that that is called as triose. So erythrose and triose, the difference in all of these, both these structure is the arrangement of OH group. So if you go back and you look at erythros, the OH group in erythros was here and hydrogen was here. Whereas in this trios, OH group on this is on the left side and hydrogen is on the right side. So the arrangement of atoms in carbohydrates is very important. This arrangement would change the type of sugar that we have. Okay, for example, question could be simple. Identify an aldo or keto and as tetros, pentos, or hexose for these sugar. For example, this is how many carbons? 1, 2, 3, 4, 5, 6. So this is a hexose. And the group that is present here is aldehyde. So this is aldohexose. This is 1, 2, 3, 4, 5. So this is a pentose. 5 is pent. And because this has a ketone group, this is a ketopentose. So it's very easy to recognize what type of sugar you have. Now another common thing, like I told you, we saw the difference in previous two sugars was arrangement of atoms. So that is why one very important type of isomer that you see in these carbohydrates and most of biological molecules are called as structural isomers. These are the molecules where the molecules have the same molecular formula but different bonding arrangements, meaning they have the same molecular formula. Example, you can see here C2H6O. This is ethanol, CH3CH2OH, which is one carbon, two carbon. So two carbon, total hydrogen is 3, 4, 5, 6. So C2H6 and one oxygen. But at the same time, you have this dimethyl ether, which is 1 and 2, two carbons. Total hydrogen is 3 and 3, 6, and one oxygen, which also gives you C2H6O. So what is the difference between these two structures is the arrangement of atoms. So when the formula is same and the only difference is in arrangement of atoms, those are called as structural isomers. Type of isomers that usually exist, and these isomers are called as stereoisomers. So what are stereoisomers? They have the same molecular formula, they have the same structural formula. Structural formula means that they have the same bonding of atoms, but what's different in them is the arrangement of these atoms in space, meaning they have the same molecular formula, they have the same structural formula, they have the same type of bonds between the atoms. For example, if we look back at the previous example that we saw, what is different between them is the arrangement of atoms, right? In the form that their structures are different. This is CH3, carbon is bonded to 3 hydrogen and it is bonded to CH2. So these bonds, here the carbon is bonded to another carbon, whereas in this case carbon is bonded to an oxygen, then it is bonded again to a carbon. So the bond arrangement is completely different. Okay, what is the difference between these two molecules? The arrangement of molecules and the bonding of the atoms. The atoms are bonded to completely different atoms in both of these structures. Okay, so these are structural isomers. Whereas in case of stereoisomers, they will be bonded to the same atom at the same time, but the arrangement in the space will be different. Okay, what that means is, for example, in this case, carbon is bonded to this H and it's bonded to this OH, it's bonded to this Cl and this Br. They all will be bonded in the exact same manner, but what will happen? I might just move around this. I'll keep this Br here, but I'll move this OH on the left. I'll move it to the right, and hydrogen, which was on the right, I'll move it to the left. Now, what have I done? I have not changed the bonding pattern. The bonds are still the same. The carbon is bonded to OH, Cl, hydrogen, Br. Still the same bonding. But the arrangement of atom in space has changed. So these are called as stereoisomers. What is important for anything to be, for two molecules to be stereoisomers of each other is two things. They have to be mirror images of each other. First thing. Second thing, these mirror images have to be non-superimposable. I'll explain what that means. And the last thing is this, there should be at least one chiral carbon in the molecule. What exactly is a chiral molecule? A chiral molecule is basically a molecule where the number of atoms are exactly same between the two molecules. The number of atoms, the type of atom exactly is same, but the structure is also same. Only difference thing is in the arrangement of atoms in the space. Chiral molecule will always have a non-superimposable mirror image. What that means is that if you put that molecule in front of a mirror, the image that it creates will be non-superimposable. Let's try and understand that with an easier example first. So for example, if you take your left hand, if I put it in front of a mirror, that is going to give me an image how this small pinky finger is going to be right here, then the bigger finger, then the bigger finger, then the index, and then your thumb on the side, right? That's how your mirror image would look like. So it would look exactly like your, like this. So this is your pinky finger, then this, then this. It looks, this is your mirror image, and it would look exactly like your right hand. Now these are two mirror images, okay, of this molecule. This is a mirror image. Now what does non-superimposable mean? It literally means that if I take this particular image from here, I lift it up and I put it on top of this left hand, how would it look like? It would look like this. This would be your thumb, this would be my index, then that, then this, then the tiny finger, oops, pinky finger, oh my god, then the pinky finger, and that would be your hand. If you literally take this and put it on top of this, what this means is that these two images are not going to hide each other. The top image is not going to hide the image behind it. Why? Because they are not, they're not same. Okay, that is what we call as non-superimposable. When one of the image, if you lift it up, you could rotate this image in any way, shape, form that you want, but there's no way that you can get it to look exactly like your previous image. Okay, that is called as non-superimposable. So your left and your right hand essentially are non-superimposable mirror images of each other if you place them in front of each other like this. So these are chiral molecules. Chiral molecules essentially are the molecules, two molecules which are basically mirror images of each other. So if you put them in front of each other, they'll look exactly like mirror images. But if you try and take one of those molecules and put it on top of the other, they will never hide each other. Those are called as chiral molecules. Okay, if those mirror images are identical and they are superimposable, meaning you could take both of these images, image 1 and image 2, and they are hiding each other, meaning they are superimposable, those molecules are called as achiral or achiral, which means they are not chiral. Okay, there's also something that is called as chiral atoms. What are chiral atoms? For example, we'll take example of carbon here because that's what we are majorly interested in. Molecules or the chiral molecules, they consist of a chiral carbon atom, first thing, and their images are non-superimposable. What is a chiral atom? A chiral carbon atom is any carbon atom that is bonded to four different groups. Meaning if you take a carbon atom and it is bonded to, let's say, hydrogen here, fluorine, bromine, and iodine. This carbon is bonded to four different groups on four different bonds. This is called as a chiral carbon atom. Most of the organic compound consist of a lot of chiral carbon atoms, and that is why we are interested in them. Why? Because they make a lot of difference. For example, let's try and understand. So if this carbon atom is chiral and this molecule is chiral, how? If you try and make a mirror image of this would be how the image look like. Carbon, the hydrogen would be right here, then we would have bromine here, fluorine here, and iodine here. Now if you take this particular image on the right side, you lift it up and you put it on top of this is never going to hide each other. Okay, for example, if I draw it right here, the carbon would be hiding each other, hydrogen would be on this side, iodine would hide itself, would be right here, bromine would be on this side, whereas fluorine will be on this side. So these two images, this image and this previous image, they're never hiding each other. That means they are non-superimposable, and this molecule is chiral, and this atom is a chiral carbon atom. So it's very easy to recognize atoms will consist of, they will be bonded to 4 different groups. Molecules is a little difficult to recognize. It is important to know that the images should be non-superimposable. First thing, they should be mirror images of each other. Second thing, they should be non-superimposable. Why do we care about this? Because these two molecules, as soon as you introduce this type of chirality into molecules, these two molecules are called as stereoisomers of each other. They have different chemical properties. They react differently with different compounds. Their chemical properties slightly change just because of the arrangement of these atoms around the space. Where I'll some easier examples to understand chirality is, for example, these two gloves. If you try and put these gloves and you imagine that there is a mirror in between, you can see these are basically spitting mirror images of each other, right? But now if you take this image and you lift it up and you put it on top of this image, they're not going to hide each other. Why? Because this thumb is on this side. Okay, so these two images are not hiding each other, which means these are non-superimposable. And these are mirror images. That is why they are chiral. If you take these two glasses, they look exactly same, yes. So if you look at this, they are mirror images of each other. But if you lift this up and you put it on top of this, it's going to completely hide it, the second one. So this is superimposable. They are mirror images, but they are superimposable mirror images, which means they can hide each other completely. Therefore, they are achiral. Now, if you look at your two shoes, this is my left and right shoe. And if I draw a mirror in between them, you can see these are very good mirror images of each other. So yes, they are mirror images. And if you lift this shoe and you put it on top of the other, it's not going to match up. They're not going to hide each other. So these are also non-superimposable. At the same time, these are mirror images and non-superimposable, which means they are chiral. So that's how you can recognize if a molecule is chiral or not. There are two important criteria. First, they have to be mirror images of each other. Second, they have to be non-superimposable. Example is right here, like I showed you. If you have this carbon, hydrogen, Bri, Cl, and you draw a mirror, it will give you Cl here, Br will be on this side, I will be here, H will be here. If you put them on top of each other, the bromine and chlorine are not going to hide each other. That is why these are chiral molecules, and these are stereoisomers. When you have these chiral molecules that are like this, they are called as enantiomers. These two stereoisomers are called as enantiomers of each other, which means this image and this image, these are two different molecules. They behave differently, they have different chemical properties from each other, and these are called as enantiomers of each other. Carbohydrates have a lot of enantiomers. Okay, for example, if we look at this, identify each as chiral or achiral compound. If we look at this molecule, carbon is bonded to hydrogen, chlorine, CH3, and this CH2, CH3. All four are different, which means this is chiral. In this case, carbon is bonded to two hydrogens, one chlorine. So these two groups are same, which means this is achiral. They have to have at least one carbon, which is bonded to 4 different atoms. Only then the compound will be chiral. Carbon bonded to all 4, which means this is chiral carbon atom. So compound could also be So let's try a question. Indicate whether each pair is a mirror image that cannot be superimposed, or are these enantiomers or not? Simple question. So if I take these two, easiest way is first draw or imagine there's a mirror in between them, a better and straighter mirror than what I drew. But oh, that looks worse. Imagine that there is a straight looking mirror between these two. Now first thing you want to check are these mirror images. So CH3, CH3, carbon, carbon, hydrogen, hydrogen here, Br, Br, CH2OH, which means yes, these are mirror images. So that criteria is good. The second is, are these superimposable or not? So if I take this and I move it directly on top of this, CH2OH and Br will hide each other, but CH3 will come here, hydrogen will come here, which means they are not going to hide. So these are non-superimposable, which means these are enantiomers. So both the criteria should meet. Only then you can say these are enantio, not meters, enantiomers. Okay, next is this. So first thing we'll do is draw a mirror in between them and then see are these images CH3, CH3, Cl, Cl, hydrogen, hydrogen. Yes, these are mirror images. But first thing, are these even chiral? There are three criterias. One is they should be chiral carbon. At least one carbon should be chiral. They should be mirror images. And then they should be non-superimposable. Okay, so in this case, these are not chiral carbon, which means these are not chiral, which means they are not enantiomers. So what is important is to see if these three criteria meet up or not. Now let's come back to sugar. Okay, like I said, chirality is very important to understand because lot of carbons in carbohydrates are chiral and that imparts them different properties. That is why we are interested in them. Now let's actually go back to sugars. First thing we'll look into sugars is the Fisher projection. What is Fisher projection? These are Fisher projections where you draw these carbons in this long format. All the carbons are connected to each other. These are basically carbons in these corners. Each of this corner is basically one carbon atom. So there's one carbon here, carbon here, carbon, carbon, carbon. And you can see there's at least one carbonyl and rest have all OH on them. So this is 6 carbon sugar called glucose, 6 carbon sugar called galactose, 6 carbon sugar called fructose. What's the difference between them is again arrangement of these atoms. A Fisher projection is basically commonly used to represent carbs. The most oxidized group is put on top, for example, the carbonyl carbon. Shows the chiral carbons as intersection of vertical and horizontal lines. So all these carbons are chiral carbon. How do you know? If you take a look at this carbon, one is hydrogen, second is OH, third is CHO, and fourth is all of this, which means it has four groups on four different sides, which means this is chiral. Same thing here. This has hydrogen, oxygen, this whole thing, and this whole thing on the other end, which means this is also chiral. So all these carbons in carbohydrates are usually chiral. So it has one chiral, second chiral, third chiral, fourth chiral, four chiral carbon atoms in this particular sugar. CH2OH is not chiral. Why? Because this carbon is bonded to two hydrogen atoms, which are same groups. Okay, so this is basically a Fisher projection where you show CH2OH in the very bottom, carbonyl on the topper part, okay, and then you're showing all of these middle carbons which are chiral with a OH and a hydrogen. Next, let's talk about D and L notation. What are these D and L notations? These are the two enantiomers that we talked about. So enantiomers Each molecule, if it has enantiomers, those molecules are called as D and L enantiomers of each other. They are stereoisomers. One is called as D, another molecule is called as L. How do you know which one is what? To understand which one is D and which one is L, we look at the chiral carbon which is farthest from the carbonyl. So when you draw the structure like this of a carbohydrate, and this is your carbonyl carbon, let's say this is my CHO, you're looking at the carbon which is chiral and which is farthest away from the carbonyl carbon. We look at this carbon and we look at where is the OH group located on this carbon. If the OH is rotated, is on the right side, it is called as a D. The D letter is assigned to the structure with OH on the right, and L is assigned to the structure with OH on the left. It's very easy to remember L. L is the one where OH is on the left, and D is the one where OH is on the right. But you remember, you're looking at the carbonyl, which is farthest away from the carbonyl carbon. The chiral carbon, which is farthest away from the carbonyl. Those are called as D and L enantiomers. Example we can see here, this is erythrose. Carbonyl carbon is this. We are looking for chiral carbon farthest away, which is, this is also chiral, this is also chiral. Both these carbons are chiral. One, oh, one and two. We are looking at the one which is farthest away, which means this. So I'm interested in looking at where is the OH. OH is on the left, so this is called L isomer, L erythrose. This one, 2. This carbon is the farthest away and it is on the right, so this is called as D-erythrose. So you can see what is the difference between these two. The only difference between these two is essentially the OH group. And if you draw a mirror in between, you can see that these are mirror images of each other. First thing, these are non-superimposable, which means these are enantiomers. And we have two types of enantiomers. One is D, another is L. D is the one which has OH on the right. L is the one which has OH on the left. And we are looking at the carbon which is farthest away from the carbonyl. Now, these two enantiomers behave differently, and you will find that in nature, most of the enantiomers that are commonly found are D. So most of the carbohydrates that naturally exist in nature are D enantiomers. Some more examples you can see here. The question could be simply, I can give you this structure, I can draw this structure in the question, and I can ask you, this is a structure of ribose. Which type of enantiomer is it? D, L cannot be determined. How can you tell it? You will look at the carbonyl carbon, which is here. Then you will look at the chiral carbon, which is farthest away from it. So this is one chiral, second chiral, third chiral. This is farthest away. So we are interested in this carbon. OH is located on the left, so this is L. Therefore, this is L. Same thing here, this is D. So this is L ribose, D ribose. They both are ribose, only difference is the structural stereoisomers of each other. Similarly, we have glucose. The farthest away carbon is this. So this is L glucose, this is D glucose. Identify the following Fisher projection D or L. Remember, all these carbons that are shown as these crisscross, these are all chiral carbons. So you don't have to worry and figure out, is this chiral or not? They are always chiral carbons. This is carbonyl. The farthest one is this. OH is on the right, so this is D. This is carbonyl. Farthest is this. OH is on the left, so this is L. That's it. This is a structure of mannose. Identify, is it L, D or L? Again, carbonyl. Farthest one is this on the right, so this is D mannose. Okay, in this case, the question is identify the number of chiral center. This carbonyl is not chiral. This is not chiral because it has two hydrogen. This is chiral, this is chiral. So total chiral centers are two. Is it D or L? You look at the farthest one. We don't care about this. We only care about this, which means OH is on the left, so this is L isomer. Now let's look at some of the common sugars. Glucose. Some things that you should remember is, what is D-glucose? D-glucose, D-fructose. Again, like I said, we are majorly interested in these. Why? Because these are more prominent ones that exist in nature. Glucose is what? It's one of the common sugars that is found in all the fruits, syrup, honey, right? What do you need to remember here is that D-glucose is an aldohexose, meaning it has aldehyde group, 6 carbons, and the formula will be what? CnH2O, which means N is hexose, so N is 6. The formula will be C6H 12O6. That is the formula of glucose. This is also known as dextrose and blood sugar. The sugar that is found in our blood, in our body, is basically called as glucose, or it is also sometimes referred to as dextrose. D-glucose is building block for multiple types of sugar that we use. For example, you might have heard of lactose, maltose, sucrose. Sucrose is the table sugar, cellulose, glycogen, all these are made from glucose. D-fructose, another common sugar which is also obtained from sucrose. This is a ketohexose. So remember this, it's a ketohexose, meaning it has a ketone group and it's hexose, so again it's 6 carbon. So what will be the formula? Formula will be same, C6H12O6. You can see their molecular formula is same, but this is an aldose, this is a ketose. Completely different structure of atoms. So these two are structural isomers of each other. As long as their structures are different, they are structural isomers. If only differences is the arrangement of atom in space, they are stereoisomers. This is one of the sweetest carbohydrates that can exist. It is twice as sweet as sucrose. What is sucrose? Sucrose is the table sugar that we usually consume regularly. That is sucrose. So fructose is twice the amount of sweetness than table sugar. Next monosaccharide example is galactose. What is galactose? It's an aldohexose. Again, it's a hexose. So all of these sugars have same formula. They are 6 carbon. Okay. But what is the difference between them? If you look at glucose, glucose is also aldohexose, which means it has the same kind of arrangement of atoms, right? But what is the difference here? It has similar structure to glucose except for OH on the carbon number 4. So this is 1, 2, 3, 4. Carbon number 4, when OH is on the left side, it is galactose. When OH is on the right side, it is glucose. So that is the difference between them. So it's very important that you know where is the OH located in order to be able to recognize what type of sugar it is. Question could simply be, draw the projection of D fructose or L fructose. So we know fructose is what? Fructose is a keto hexose. Hexose means 6 carbons. So first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and sixth carbon. What is important here to remember is, oh, I'm not able to. What is important here to remember is that it's a keto, meaning the second carbon here is going to be a ketone group. So it would have C double bond O. Now the rest you just add hydrogen. So this should have an OH group. 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 And that's 1, 2, 3, 4, 5, 6. CH2OH, right? But you cannot just draw OH wherever you want. OH have a specific spot. D tells you that the last carbon, this is carbonyl, so last carbon here should be on the OH, should be on the right side for the D. But what about D's? This is something that you need to memorize. For example, when you look at this is fructose. When you look at fructose, carbon number 1 has OH on the left. Rest all are on the right, and this is D. If you move this also on the left, then that would be L fructose. Okay, so D and L you, as long as you can memorize one, the other one is basically mirror image of it. Okay, so D has this OH on the left, and then you just draw a mirror image of it, and that would give you the L fructose. Okay, next thing is in case of carbohydrates, one another important thing is Haworth projection. So most of the carbohydrates, they are not stable when you draw them like this. This is not usually the stable form of carbohydrates. Carbohydrates usually are more stable, especially 5 carbon and 6 carbon sugars are more stable when they form a ring structure like this. So monosaccharides usually will form a ring structure, and these ring structures are called as Haworth projection. You can see this is also 6 carbon. 1, 2, 3, 4, 5, 6. This is also 6 carbon. Only difference here is it has formed a ring structure now. Okay. Less than 1% of the sugars are found in the open format. Open chain meaning the Fisher projection that we saw. Most stable form are these 5 ring structure or 6 atom ring structure. They always have to be arranged in rings like this. What are these? These are cyclic hemiacetols that are produced by reaction of carbonyl group and the hydroxyl group in the same molecule. Okay, and they have a different type of isomer present in them. These isomers are called as anomers. When you draw Fisher, we have enantiomers. When you draw Haworth, the isomers are called as anomers. So let's look at how are these enantiomers formed, Haworth projections formed. You don't have to know exactly how to draw them, but you should at least have an idea how these are formed. So I'm just going to explain quickly, briefly, how are these formed. So if you remember from the chapter of aldehydes, aldehydes react with hydroxyl group. They always react, they can easily react with each other. What is happening in case of a glucose or these 5 or 6 carbon sugars? They usually have a carbonyl group and they have bunch of OH group in the same molecule. So this OH, this carbonyl here and the OH here, they are going to react with each other in the same molecule. How? This is exactly how. So if you draw a chain structure, the carbon here, the OH from this 5th carbon is going to interact with the carbonyl carbon, and the structure is basically going to bend on itself. See here, this is the same 6 carbon structure. It's just that it is now in a bent format. Okay, this is carbon 1, which is carbonyl. 2, 3, 4, 5, 6. 6 carbons. I just simply bent this structure. Why? Because this carbonyl is now going to interact with this OH. Okay, how? This hydrogen will be lost. If this hydrogen is lost, that means this oxygen will need one extra hand. Similarly, this bond, one of these bonds will be broken and this will be lost. Oops. This will be lost, because of which this oxygen is going to make a bond with this carbon directly. Okay, so this oxygen will make a bond with this carbon just like this. And that is how you get this particular structure, which is a 6 carbon ring, 6 6 atom ring, where 5 of those atoms are carbon. So this is carbon 1, 2, 3, 4, 5, and this 6th atom is an oxygen. It's a 6 atom ring, but one of those atoms is oxygen, and this is your 6th carbon. This is carbon number 1, carbon number 2, carbon number 3, 4, 5, and 6. It's a 6 carbon structure, but the ring is basically 6 atoms, and one of those atoms is oxygen. Okay, so this is how glucose Haworth projection is formed, and this is how most of the glucose is found in nature. It's a ring structure which is more stable than having that long chain of carbons. That is not very stable. Now, we talked about isomers. Whenever you have Haworth projection, the isomers in this case are not called as D and L enantiomers. They are called as anomers of each other. Oh, sorry, anomers. They are called as anomers of each other. What are these anomers? To understand anom, Isomers are the monosaccharides that are again stereoisomers of each other. And how do they differ? They differ in configuration only at their anomeric carbon. And what is the anomeric carbon? The carbon which is coming from carbonyl. So remember, this carbon right here was the carbonyl carbon previously, before it formed a ring structure. So the carbon was a part of, which was a carbonyl, is called as the anomeric carbon. An easy way to remember it is, if it is, the structure is a six-membered ring, it's usually this first carbon. That's the anomeric carbon. And the two isomers that it forms, they are called as alpha and beta isomers. Okay, so in enantiomers, they were called as D and L. In case of anomeric carbons, they are called as alpha and beta. How? Alpha means that the OH group on this anomeric carbon is going to be written below the plane. They would be facing down. If it is written down, it is alpha. If it is written up, it is beta. That's it. So D and L they were on the left or the right. Okay, left was L, right was D. In case of anomers, if the OH is on the bottom, it is alpha. If it is on the top, it is beta. Okay. So for example, this is an alpha D glucose, meaning it's a D glucose because this is coming from a D Fischer projection. In this D, the carbonyl and the OH reacted with each other. So it is coming from a D glucose, but it is alpha. Why? Because now the OH here is located at the bottom. So that is why it is called as alpha D glucose. If this structure was coming from an L, it would be called as an L glucose, and it would be called as alpha or beta depending on if the OH is on the top or is on the bottom. Okay, now again, like I said, you have to kind of memorize these structures because there's no other way. OH somewhere is on top, somewhere is on the bottom, and this is how the structure is. If you switch this OH from top to bottom, you have a completely different structure. If you move one of the OH around, remember, you could get, instead of glucose, you would get galactose, or you would get some other sugar. So the only difference here is, it's important that you remember where is which OH located. For example, one easy way to remember in case of glucose is that it is up, down, up, down, up. Though that is how the OH are located. Example in case of beta. OH is on top, then down, then up, then down, then up. Okay, so if you remember the beta anomer, up, down, up, down, up. This is the arrangement of OH. And what would be in alpha? If you draw the beta first, alpha would only have one difference, which would be this. Only this OH is going to move, nothing else. So you can see in alpha, instead of having it up, we have this one down, and then rest everything is same. Down, down, up, down, up. Everything else is same, only difference is these two. Okay. Easy way to memorize is to memorize the beta one. Up, down, up, down, up. Okay, and this ability of the molecule to sometimes form a ring structure like this, and sometimes turn it into like a Fisher projection, like an open chain, which is this. It's not drawn like a straight chain, but basically this is a straight chain. They are not reacting with each other. This process is called mutarotation. The ability of monosaccharide unit to be in open or cyclic form. Okay, when you put this in solution, sometimes they can basically go in between each other. Some of the molecules will be in the Haworth projection, some will be open, meaning they'll turn into Fisher projection. But always remember, this is the trace amount. It's a very small amount of the structure of carbohydrates that are found in the open format. Most of them prefer to be stay in the ring structure, but they can always go from one structure to other, and this is called as mutarotation. This is a Haworth projection of fructose. Fructose is a ketohexose, which means this. Again, carbonyl carbon is going to interact with the farthest chiral carbon. So this is how many carbon? 1, 2, 3, 4. Only 4 carbon, and one of them high oxygen. That is why this ring is a 5 atom ring. When you see A 6 atom ring, that is usually a glucose, galactose kind of format. When you see a 5 atom ring, that is usually fructose. So this is fructose 1, 2, 3, 4, 5, 6. Important thing here is which carbon is anomeric. In case of fructose, the second carbon is anomeric. Why? Because it's the second carbon that usually interacts here. You can see this carbon is interacting with the OH. So in case of the 6 atom ring, carbon number 1 is anomeric. In a 5 atom ring, carbon number 2 is the anomeric carbon, which means you look at carbon number 2. If OH is on the bottom, it is alpha. If OH is on the top, it is beta. Fructose is different. Here you'll see CH2OH and CH2OH on both the sides. Okay? But again, anomeric carbon is only this OH. And how do you remember it? Down, up, down, up. Same mantra, but now you're remembering it for alpha. In case of the beta glucose, you were remembering it for beta. In case of fructose, it's the same mantra, but you're remembering it for the alpha structure. Okay. Next, we'll look at some of the chemical properties of monosaccharides. Monosaccharides can form sugar alcohols, okay, which means if you take a sugar which was aldehyde or a ketone and you reduce it, it is going to form what is called as alcohol, sugar alcohols. Example, sorbitol, xylitol, xylose, mannitol, mannose. These are all the other type of sweeteners that are used in many sugar-free products. Okay, you might have heard of this xylitol or sorbitol before. These are usually used as sugar in the sugar-free products. And what are these? These are also sweet, but they are not sugars, they are sugar alcohols. So some of the reactions of these monosaccharides or sugars are reduction and oxidation. Reduction means you have seen these reactions in the previous chapters. So when aldehyde or ketone, they can undergo oxidation. When they undergo oxidation, they form carboxylic acid. When they undergo reduction, they form alcohol. So it's the same a process, but only difference is now you have a bigger molecule. In this case, you have aldehyde here, like glucose. When this will undergo reduction, how? Reduction is when you add hydrogen in presence of a catalyst. It will undergo reduction to form alcohol. The aldehyde will be turned into alcohol. You add hydrogen into this. Okay, CHO will turn into CH2OH. This is called as sugar alcohol. This is glucose. This is called as glucitol or sorbitol. Similarly, monosaccharides can undergo oxidation. Like I showed you earlier, oxidation means aldehyde will be converted to acid. Aldehyde is CHO, acids are COOH. So how would this undergo oxidation? One of the agents that is used for oxidation is called as Benedict solution. Benedict solution or Benedict reagent. This is an oxidizing agent which will convert your aldehyde into acid. So whenever you see this O written on or you see Benedict reagent, then that is going to oxidize your aldehyde or ketone into an acid. Ketones are usually difficult to turn into acids, but aldehydes will be turned into acids. Okay, how? What are these Benedict solution? Benedict solution usually consists of copper, 2 plus ion in it, which is an oxidizing agent. Okay, and these sugars are getting reduced. So they are also called as, they are reducing this, they are getting oxidized, they are reducing the copper. So they are also called as reducing sugars. The sugars that get oxidized are called as reducing sugars, and not all sugars can be oxidized. Most of the sugars can be. One of the sugars that cannot be oxidized is sucrose, and we'll look into that later on. But basically they have copper in them, which is the copper is the Benedict reagent. It's usually blue in color. When you mix it with sugars, it turns reddish in color. Why? Because it forms copper oxide. So basically copper ion is reduced to copper oxide. Okay? And this is one of the tests that is used for carbohydrates. So carbohydrates usually, when you mix them with Benedict, they will, if they are positive test, they will turn the blue Benedict into red. Fructose is a reducing sugar because it can, under basic conditions, fructose, even though it is ketone, ketones don't get is cannot be reduced oxidized to acid, but still fructose will give you a positive test for Benedict. Why? Because this ketone group will rearrange and it will turn into an aldehyde. So fructose and glucose, remember this is also C6H12O6, this is also C6H12O6, and they keep on converting into each other. There's always a rearrangement going between these molecules. So whenever fructose will be converted to glucose, that glucose will get oxidized by Benedict reagent. So that is why even fructose gives you a positive test. Even though it shouldn't, but it still gives you a positive test for Benedict. Why? Because it can rearrange into glucose. Next thing we'll talk about are glycosidic bonds. Okay, what are glycosidic bonds? We just saw how we have seen this earlier, that aldehyde can react with alcohol and it will form something that is called hemiacetal. And what are hemiacetal? Hemiacetals are what give you Haworth projection. All the Haworth projection you saw were hemiacetals. These hemiacetals can further react with OH and that can give you acetal like this. And this bond is called as a glycosidic bond. When you see a bond like this is called as a glycosidic bond. When a monosaccharides react with alcohol, it forms a glycosidic bond. Example, you can see this sugar. If you take this sugar, this is an alcohol here, and this is another alcohol. So when these two react with each other, they can give you a bond like this, which is COC bond. COC bond, which is called a glycosidic bond. Why are these important? These glycosidic bonds are important because they make the links between monosaccharides. Remember, disaccharides and polysaccharides are formed by joining of monosaccharides. How do these monosaccharides join with each other? They join with each other by glycosidic bond. So if what is the bond, the type of bond that is found between monosaccharides? One mono, second mono, third mono, how do they join? They are joining to each other by a bond which is called as glycosidic bond. So let's look at some of these di and polysaccharides quickly. Disaccharide example is lactose. Okay. Lactose is what? Lactose is, where do you find it? You find it in milk, milk products. This is one of the sugars found in milk. It is a disaccharide which is formed by joining of two monosaccharides. What are those? Galactose and glucose. When galactose and glucose, beta D-galactose, alpha D-glucose, when they join with each other, they form what is called as lactose. What you need to remember here is the type of bond that is found in these disaccharides. Okay, so glucose and glucose makes maltose. Glucose and galactose makes lactose. Glucose and fructose makes sucrose. So ideally you should remember these names. Glucose gives maltose. Glucose galactose gives lactose. Glucose fructose gives sucrose. So make sure you know these names. What are the monosaccharides that combine and give you what disaccharides? Okay. Let's look at some of these examples. Maltose. Maltose is a disaccharide known as malt sugar. How is it formed? It is formed by two alpha D glucose molecules. When two glucose combine, which glucose? Specifically alpha D and alpha D. How? This is alpha D, alpha D. What is the type of bond formed in them? This is the bond formed. Carbon number 1 from one of the glucose, carbon number 4 from the second glucose. The OH from this and hydrogen from this will react. OH and H will make H2O, which is water. Water will be lost like here, and you will have a connection between them. So this carbon will link to this oxygen. This oxygen is already linked to this carbon, and that is how you get this bond. COC bond. This is the glycosidic bond, and this is called as alpha 1,4 glycosidic bond. Why? Because both of these glucoses are alpha, and the bond is present between carbon number 1 and carbon number 4. So make sure you know this name. What you need to remember here is you should be able to recognize the structure of maltose if you see it. Second thing, maltose is a disaccharide, remember that, made up of two glucose. What are the two components? Alpha D glucose. And second thing you need to remember is the bond, alpha 1,4 glycosidic bond. Okay, alpha means both are alpha, 1,4 means carbon number 1 and carbon number 4. Okay, lactose is another type of disaccharide formed between galactose and glucose. Specifically, which type? Beta D galactose and alpha or beta D glucose. This could be any. The second one. Second one could be alpha or could be beta, but galactose is always beta. How are they going to link? Exactly like this. This is beta D galactose, which has beta means OH on top. Alpha D glucose is this OH in the bottom, and this is how they will link carbon number 1, carbon number 4 again. So what is the linkage called? It's called beta 1,4 glycosidic bond, or it can also be called as beta alpha 1,4 glycosidic bond, depending on which ones are you linking. Okay, remember here this is beta, but this could be alpha or beta. So if this is beta and beta, it would be beta 1,4 glycosidic bond, or if this is beta and this is alpha, then it would be beta alpha 1,4 glycosidic bond. What you need to remember is the sugar, the if it's a disaccharide, monosaccharide, polysaccharide, what are the monosaccharides present in them and what type of linkage is present in them. Okay, so you should know these. Next is sucrose. Sucrose is also a disaccharide. It's the normal table sugar that we consume. It is made up of two sugars. One is glucose, another one is fructose. What type of sugars? Alpha D glucose and beta D fructose. Remember, they're mostly always going to be D's because those are more prominent. Important thing to remember is alpha or beta. So alpha glucose and beta fructose. What type of bond will they form? Carbon number 1 from glucose and carbon number 2 from fructose. So that is why the bond will be called as alpha beta 1,2 glycosidic bond. And this is how it would look like. One would be a 6 atom ring, another one would be a 5 atom ring. This is glucose, this is fructose. They make table sugar. Next, we have polysaccharides. What are polysaccharides? Polysaccharides are basically the type of sugars that consist of multiple monosaccharides connected to each other, right? So these are the monosaccharide. When you have something connected like this, multiple chains of uh, monosaccharide units, those are polysaccharides. Example, there are again a lot of examples of this as well. Example is amylose, amylopectin, cellulose, starch. Those are all examples of polysaccharides. Let's look at some of them one by one. This is an example of starch. Polysaccharides, amylose and amylopectin. What is amylose? When you have these straight chain of polysaccharides of glucose connected to each other, when they are in a straight chain like this, all these are glucose connected. One glucose, second glucose, third glucose, they are connected in a straight chain. What they make is called as amylose. The structure of amylose is a straight chain polysaccharide of glucose. Amylopectin is branched chain of glucose, meaning you see here this is glucose, glucose, glucose in a straight chain, then suddenly there's a branch, glucose, 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 and this is, there could be another one going here, another glucose, 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 and this is another one, glucose, glucose, glucose. So when there's a branch present, it's amylopectin. When it's straight, it is amylose. Okay. Again, what you need to remember here is that they are polysaccharides. What is the sugar that is involved? It's glucose. And what is the type of bond that is involved in each? Amylose has alpha 1,4, alpha 1,4, 1 and 4, 1 and 4, 1 and 4, alpha 1,4. When they are all alpha 1,4, they make amylose. When there is alpha 1,4, 1,4, one of this, four of this, one of this, four of this. But there's also one and six. So there are two types of bond. The name of bond is what you need to remember. This is only alpha 1,4, this is alpha 1,4 and alpha 1,6 glycosidic bond between sugars. So amylose only has one type of bond, amylopectin has two types of bond. Why do we care about this? Amylose and amylopectin basically make starch. What is starch? The sugar that is stored in by plants. That is starch. Amylose makes up 20% of the starch. It consists of glucose molecules. How many? 250 to 4,000 connected by alpha 1,4 glycosidic bond. What you need to remember is starch, glucose, alpha 1,4 bond, and they're all straight chain. Next is amylopectin, which makes 80% of the starch. It's branch. Again, alpha glucose. What is the bond? Alpha 1,4 and alpha 1,6 glycosidic bond. Starch is the sugar that is stored in plants. Starches hydrolyze easily in water and acid to give smaller saccharides like dextrins, which then hydrolyze to maltose and finally glucose. So we eat these starches and our body can then digest it and convert it into glucose. How does our body hydrolyze the starch? The enzyme that our body has to digest starch is amylase and maltase. Amylase is found in the saliva. So when we eat some of these carbohydrates, amylase starts digesting protein, these sugars, they turn it into dextrin. The dextrins are further digested, converted into maltose. They are further hydrolyzed by maltase and converted into glucose. Only then we can finally get glucose, which we can consume and use to produce energy in our body. Next type of polysaccharide is glycogen. Glycogen is the type of sugar that we store in our bodies, that humans can store. Okay, glycogen is a polymer of glucose that is stored in liver and muscles. So starch is the type of sugar that is stored by plants, and glycogen is the type of sugar that is stored by animals. Why do we store it? Again, for the same reason, for the times when our body is not eating food but we need to use energy. Glycogen is hydrolyzed in our cells at a rate that maintains the blood level of glucose and it provides energy between meals. It is similar to amylopectin, but it is a little more branched. Example, glucose units in glycogen are joined by alpha 1,4 and alpha 1,6 bonds as well. But in this case, there is a branch after every 10-15 units. Whereas in case of amylopectin, the branches were a little farther away from each other. Here the branches are very fast, coming out pretty fast. The next type of sugar polysaccharide is cellulose. You might have heard of cellulose. This is found in plants. It is the structural unit of plants. They're found, it's in the cells, a membrane. It forms some cell, the cell membrane, and then there is cellulose around it, right? This is a polysaccharide. How is this formed? Again, it's made out of glucose in unbranched chains. Again, this is not branch, meaning straight chain, and it consists of beta 1,4 glycosidic bonds. So what you need to remember is, again, it's monosaccharide, polysaccharide, disaccharide. If they are di or poly, what type of monosaccharides do what monosaccharides join to make them up, and what is the type of interaction? This cannot form hydrogen bond with water, that is why it is insoluble in water. This is the sugar that makes the cell walls around the cell. So the ones that I said, there's plasma membrane, then there's cell wall around the cells. In plant cells, that is cellulose. It is very resistant to hydrolysis. Starch is something we can digest, but cellulose is something that we cannot digest. Humans cannot digest. Why? Because we are not able to break this beta 1,4 glycosidic bond. Our body only has enzymes that can break alpha 1,4 bonds, but our body does not have enzymes to break beta 1,4 bonds. That is why we cannot digest cellulose. Looks like this. Cellulose, okay, beta 1,4 glycosidic bond. We cannot digest them because we do not have the enzyme for